Good evening, ladies and gentlemen, and welcome to today's lecture in the series of scientific evenings at the University of Nova Gorica. I'd like to introduce to you our speaker today, Professor Doug Saddy. The scientific and academic path of Professor Saddy is remarkable in at least two respects. One, it spans at least 3 continents. Second, it spans at least 3 or 4 scientific fields. So, Professor Saddy started his career at the Massachusetts Institute of Technology, where he got his PhD under supervision of Noam Chomsky, and then held positions in cognitive science and linguistics in Europe, Max Planck Institute of Psycholinguistics, Germany, Max Planck Institute of Psycholinguistics, and then he went to Australia, University of Queensland. After which he returned to Germany, where he was a professor of neurolinguistics at the University of Podan, and also a co-founder and director of Institute of Complex Systems Dynamics, and also the director of Institute of Linguistics. Professor Saddy currently holds a chair at in language sciences at the University of Reading, where he's also a founder and the director of the newly created Center of Integrative Neuroscience and Neurodynamics. Now, the research and teaching interests of Professor Saddy, as I said, span more than one field. His main specialization is language processing, but he takes the language processing in a larger context, and he's actually the first expert in our field who started applying methods of nonlinear systems dynamics to the signal analysis of language processing. In this sense, this is an extremely well-received view, which meets very well with the demands and challenges of current linguistics, which is gradually becoming more and more a natural science, especially in the light of recent developments in the imaging technology, which allows us to look at the brain patterns of linguistic tasks in closer and closer detail. So that boosts the field of linguistics a great deal, but it's only a handful of researchers that can handle the highly interdisciplinary task of balancing what we know about linguistics and linguistic data with the analytic tools and conceptual solutions that come from other sciences. An example of this synergy you're going to hear today. So today's talk is called Measuring Language in the Brain. Please join me in welcoming Doug. Doug, welcome to Slovenia. The floor is yours.
Thank you very much, and thank you for your kind welcome. It's an honor to be here. I'm really, really pleased. It's really my first visit to this country, although I did mention I passed through here accidentally in 1976, and I didn't stay very long. That was in a previous and less auspicious mode of life. Arthur asked me if I'd come and talk, and we talked a bit about the kinds of things that I could discuss. What I'm going to try to do today is really a Cook's tour of research that I've been involved in that relates to the problem of trying to connect the notion of language as an object of study with the desire to understand the workings and mechanisms, architecture of the brain. If any of you know anything about any of the topics here, you will be disappointed. I'm going to be touching lightly on a lot of things, but I'd be glad to discuss anything in questions. I'm going to try and stay within the time limit. Arthur's promised to wave at me when I'm getting close to the end of my time. I'll leave time for questions. I guess given the nature of the venue, I should wait for questions at the end. But actually, I don't care. If someone feels really strongly about something I say, just jump up. Okay, well, just to put things in historical perspective, we've been at this task for quite a while. The first written record that talks about the brain and its physical structures is in the Edwin Smith surgical papyrus, which was written around 1700 BC, but we know draws on texts from at least 3000 BC. So when we talk about the idea of understanding the structures of the brain, there's a fairly long history. And you get to, I know that my daughters, for instance, know about Imhotep because there's these movies, The Mummy, and I think Imhotep is the bad guy in them. Where's that thing? That's unfair. I sort of provocatively use the term measuring language because language is not the kind of thing that you would imagine you can measure, right? So, but of course, we're trying to do some type of empirical investigation, so measurement is what we're about. So we need to sort of address the question of why you'd be interested in this topic in the first place. Oh, I've got one of these buttons. Well, one of the reasons to address the issue of mind and language is because people care about the mind-brain interface. And generally speaking, if you scratch any of the people working in this domain, they tell you that that's the kind of thing that they're really interested in, that they want to know if everything can really be reduced to physical principles or not. So addressing the mind-brain interface is one reason you might want to take a look at the relationship between language and the brain. Why would you focus on language? Well, one of the good reasons for focusing on language is that language is a universal human trait. So if we're interested in some aspect of humanity and the brain, language is a good candidate for things to look at. While we share a lot of properties of language with other species, the human unique ability to communicate in fora like this set us apart. Another reason to think about it is that language is relatively circumscribed. When I say language to you, we all pretty much agree on what it means. It's not difficult to set the boundaries of what constitutes language. There's some fuzzy edges, but they're not really hard. Whatever properties language has to reflect the cognitive architecture. That is, we know that if we take the brain out of Arthur will stop speaking. So we could put some machines on to keep his heart going and his lungs going, but he definitely won't talk. So we know that whatever it is that supports language has got something to do with what's between our ears, and presumably the properties of language, the sort of structures of language that we find, the phenomena that we see associated with language, will tell us, at least indirectly, something about those properties of the brain itself. And finally, there's a long tradition of research into the neurological correlates of language, going back to the Egyptians, more recently going back to research done shortly after the Second World War, where mankind managed to do something that we can't do ethically as researchers, and that is make a number of very interesting brain preparations that have very discrete, neat holes in them. So people coming back from the First and Second World War with gunshot wounds to the brain actually provided us with an opportunity to look at cognitive deficits which seem to be correlated with damage to the brain in a unique way. And in fact, the explosion of interest in what was called neuropsychology really fell after that period where, because of sulfa drugs and then antibiotics, people weren't dying from these wounds. So we now had people who survived catastrophic lesions had circumscribed holes in their brains and behaved in particular ways. And then finally, a reason to approach it this way is that we've got another independent research discipline which is aimed at describing human language. And this is the one reason why language studies actually played a seminal role in the development of cognitive science as a discipline that we're familiar with now. Language studies allow us, or linguistics or linguistic description, allow us to have paradigms that have a vocabulary and a set of explanatory profiles which have been established independently of the kinds of experiments that we're doing. Right? So we aren't developing a vocabulary or theory of language based on our brain investigations. We actually already have a theory of language, and the question is whether or not we can connect these things together. All right, so what kinds of things would we need to know? Well, we need to know something about the general components of cognition if we're going to address this linkage between language and the brain. So that is, what other cognitive phenomena do we have to assume are there in order to account for the existence of language? We would need to know something about the relevant properties of language that we would like to investigate, so which aspects of language are worth looking at, and which aspects of language are perhaps less interesting from the perspective of understanding the linkage between language and the brain. We can ask, what is the basic nature of the neural computation? So what kinds of things are the brain doing such that it can do language? And is there anything special about that? What neuroanatomical areas of interest exist? So are there particular areas of the brain that are crucial for language? Are there particular areas of brains that are of particular interest, whether or not they have anything to do with language? Well, that series of questions basically defines an enormous research agenda, right, with which cognitive science is sort of the overall label that takes these things into account. When we ask the question, what relevant properties of language do we have to investigate? Well, we can ask questions specifically about the languages themselves, but we can also ask questions about just the phenomena that we see when we look at language. We know that language processing is necessarily incremental. It has to unfold in real time, right? I start to articulate, and it takes time for noise to come out of my mouth, so I've got to have some planning organism inside me that allows me to get ready to move my articulators and to make noise that I think you will understand. And you're going to hear my expression come to you in real time, and it'll come in a sequential way, and you're going to have to recognize and unpack that noise in such a fashion that you can understand what I'm talking about, hopefully. If we look at speakers of English, I couldn't get the data for Slovenian, but we could probably find it. Speakers of English speak at a rate of about 3 words per second, sometimes a lot faster. So if I'm one of those, you can wave and slow me down. Speech errors occur at a rate of approximately 1 per 1,000, which is not a bad error rate. And comprehension rates are much faster, and the error rates in comprehension are lower. Now, how to actually explain that set of observations is a real challenge. We can't produce computer models that anywhere near approach these levels. So how is it possible that language can be deployed in real time? Not only do we have the challenge of trying to account for the rate at which we can deploy language and understand it, but we actually have problems that are associated with the language themselves. So let's look at a few things that are kind of make it kind of tricky to explain how language can really be deployed. Here's a good sentence. This is a sentence that if you've studied linguistics in the sort of Western-speaking world, you will have encountered it. If you haven't, and you're a native speaker of English, you'll think it's a bad sentence. If you haven't, and you're not a native speaker of English, bear with me and I'll try to explain to you why it's a weird sentence. Okay. The horse race past the barn fell is a kind of sentence that naive speakers of English will reject when they're first exposed to it. They'll simply say, this is not an appropriate sentence of my language, I don't understand it. Actually, it's a perfectly good sentence of English. English allows something called reduced relative clauses. The horse raced past the barn fell is another way of saying the horse that was raced past the barn fell. Okay. But people don't notice that when they're first exposed to it, particularly when it's presented in a visual form. And that tells us that although as speakers of English we know about the properties of reduced relative clauses in our language, we don't use that knowledge at the time we're initially exposed to such sentences, and we're unable to determine a constituent analysis that makes sense. Then we've got an alternative, which is sentences like this one. More people have been to Berlin than I have. This is a sentence where native speakers of English will tell you it's a perfectly good sentence of their language until you ask them what it means. And then, generally speaking, you get three different views of what it might mean, but ultimately it always falls down. So here's an example of something where native speakers think it's okay, but actually it turns out not to be a possible expression in their language. And so here we have a case where we're unable to assign an interpretation. Then if you speak a really inflectionally impoverished language like English, you will encounter sentences that look like this one. So believe it or not, this is a sentence. Okay. And it's quite easy to explain to people who've been around the block for a while. So we have police, and what do? Police. Police. Okay, but if the police are doing the policing, who is policing the police? Well, police. And who polices those guys? Well, the police the police. Okay. So we've got ambiguity all over the place. Okay? English is a pathetic language. Right? You can get away with this type of stuff. And then we also have expressions like this. So my sister's new boyfriend is a louse. In fact, all of her boyfriends have been lice. This is not a particularly nice thing to say in English, and it's certainly not what one would intend to say also. So the plural of louse in English is lice. It's an irregular plural form. But when you use the irregular plural form this way, it can only mean little crawly creature. When I say my sister's boyfriend is a louse, I mean my sister's boyfriend is somebody I don't particularly care for. In fact, all of her boyfriends have been people I don't really care for. To say that, I would have to say louses, use the regular plural form, which actually isn't legal in English, except in this particular type of occasion. So there's context dependency all over the place. We've got context dependency, right, where the choice of whether you use the irregular plural form depends on whether you're using the word louse in its literal sense or in sort of what's the word? Figuratively. Thank you very much. We've got ambiguity between whether something's a noun or a verb. Here we've got a situation where you think it's okay, but you can't actually understand it. And here you don't like it, but it's actually okay. So given this phenomena, how is it possible that we can actually understand and produce language at the rates that we can? Well, we have some ideas about that. We think that the complexity in natural language comes from two distinct sources. Computational complexity, which is inherent in the grammar of the language itself. Okay, so the grammar of English, the grammar of Slovenian, will have properties which will actually determine certain aspects of how the language functions and that give it an inherent complexity. But there's also procedural complexity which comes about from marshalling processes and psychological processes in order to be able to deploy that knowledge and that information. Okay. So we need to have things like short-term memory, etc. And distinguishing between these two sources of complexity is one of the things that we hope to be able to do by looking at the brain and how it works. So we've got this general picture characterizing language use system, right? How we use language is there's some set of information that's stored, which gives us linguistic information, lexical entries with phonological, syntactic, semantic, morphological rules. as well as information about the lexical entries themselves, and a set of psychological resources like short-term and long-term memory, auditory and visual analysis routines, lexical access, and parsing routines. So there's another set of machinery that we bring to bear. When we try to approach these issues, then we've got sort of two pots. We've got a pot that says there's these psychological resources and machines that we need to understand and try and locate in the brain and understand what underpins their function and their application. And then we've got this set of linguistic information. We want to understand that as well. And of course, this is the job of linguists. And there's a bunch of different approaches, right? There's a variety of different ways in which language theorists have approached the task of explaining the kind of knowledge that we have in our brain. And so there are different theories. And the different theories make different claims about the nature of linguistic knowledge. If we had a room full of linguists, we could have a really big fight about the relative merits of these things. The reason that this is important to note is that depending on the theory that you're assuming, the nature of your description and the nature of your explanation and the nature of your vocabulary will change. Okay? So attempting to make very, very fine-grained claims about the relationship between language and the brain is going to depend not only on our ability to look at the brain and understand it, but also on what type of descriptive approach and what type of theoretical framework we're adopting with respect to the nature of language itself. Okay, so that's the setup. I've just convinced you that the whole enterprise is impossible. And now we'll talk a bit about some of the neuropsychological evidence. These two pictures here are to remind me to tell you that we care about sort of the structural properties of the brain, but we also, which here, this is short and long-term cortical fibers modeled, and we also care about the blood supply to the brain. The neuropsychological evidence came about mostly by, as I said, evidence from gunshot wounds, and then also evidence from people who've suffered strokes. And a stroke is an interruption to the blood supply to the brain, which then arbitrarily knocks off chunks of function. Okay, so we go back to the Egyptians. In the Edmund Smith surgical papyrus, there's case 22, the fracture of the temporal bone of the skull. The patient was unable to speak, and this is thought to be the first documented case of a language impairment subsequent to brain damage. This was actually an interesting case. This was a scribe who worked during the building of the pyramids, and he suffered a blow to the head and subsequently could no longer work as a scribe, not because he couldn't chisel his letters, but because he no longer had command of his language. This particular observation then predates the classically attested initial description of language impairment as being associated with damage to the brain, which we attribute to a guy named Paul Broca, who gave an address to the French Academy of Sciences in 1861, where he pointed out there was a link between the physiology of this particular brain. So Broca was seeing a guy named Tan. He was called Tan because that's all he could say. He said Tan, 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 Tan. And Broca believed that there was this link between the structures of the brain and the structures of the mind. And he, after Tan died, he did an autopsy and saw that there was damage in this particular region of Tan's brain and made the claim that there was actually a causal linkage between damage in this region and the fact that this person had lost his ability to speak. This caused quite a bit of excitement at the time. This Frenchman claiming to have found an area in the brain that was linked to language, so the Germans ran off and came up with another area as quickly as possible. And so Carl Wernicke also described an area of the brain which seemed to be linked to language production. Damage to Wernicke's area caused also a problem in language processing. And so we have this sort of classical model which says that there's a linkage between Broca's area, Wernicke's area, and some conceptual structure. And the sort of taxonomy of language impairments that we still have with us is based on whether or not there's damage to Broca's area, damage to Wernicke's area, damage to something that connects the two, and damage to how those areas themselves are connected to conceptual structure. This came out of research in the 19th century that included Broca, Wernicke, Lichtheim, and Freud, who was not generally associated with aphasia research, but wrote a particularly important monograph on language impairment. Okay, so what are the neuroanatomical areas of interest? Well, we've got what's called the Wernicke-Geschwind model. And this is the clinical classical description that there's in the brain an area here, which is in the prefrontal cortex above the temporal lobe on the central sulcus, which is Broca's area. That there's an area here in the interface between the temporal lobe and the parietal lobe called Wernicke's area, that there's some kind of connection between the two. We've got primary auditory cortex, which is in the temporal lobe on the flat part in there, and this arcuate fasciculus is the thing that connects them. The motor cortex is on this side of the central sulcus, and that's what has to be engaged in order to make your lips and tongue move. So given this is Broca's area, this is Wernicke's area, here's the connection between the two, here's the region for sort of conceptual structure making in the classical model. Then we have something like repeating a spoken word, according to this model, requires us to first of all bring in information into the primary auditory cortex, transfer that information to Wernicke's area, which then communicates it to Broca's area, which then tells the articulators what to do. To read a word, similar pattern, except that now we engage primary visual cortex. So we have to be able to see it. Information is sent from the visual cortex forward into this associative area that includes Wernicke's area. It then sends information to Broca's area. Broca's area then engages the motor cortex. So this is the classical model of the neuroanatomical areas of interest, and it hasn't really changed much. What has changed is that we now know that these areas, while they are indeed engaged in language use, are a tiny, tiny subset of the brain areas that are engaged. So we've got new brain imaging technologies, and we can watch the brain doing things in healthy condition. We don't have to have people who have suffered strokes or gunshot wounds. We can look at them while they're doing language processing and use these new technologies. The technologies allow us to non-invasively, that means we don't have to drill holes in their head, look at the brain at the microscopic level. That's actually fairly recent with the advent of very high magnetic field scanners. At the macroscopic level, which is the conventional MRI scanning technology, which we'll talk about, and also at the mesoscopic level, which is something in between. So we need to pause and give some thought to the overall dynamic organization of the brain. We've got three different spatial scales: the cellular, the mesoscopic, and the global. So it's individual neurons, big clusters of neurons that act coherently, and the whole brain, more or less. And we've got different time scales. We've got slow responses and fast responses. Neuron-to-neuron interactions take place at the time range of tens of milliseconds. The metabolic and hemodynamic response to the brain as an organ as a whole takes place at the range of about seconds. And the mesoscopic interactions, this is the interactions of fields of neurons interacting with other fields of neurons, which is evidenced by things like phase coupling in the brain, are paradoxically sometimes instantaneous. So this has been, well, it's not paradoxical, but it used to be described as paradoxical. So you would think, okay, you've got some kind of linear relationship. It takes some time for neuron A to talk to neuron B, and the time scale there is in tens of milliseconds. It takes some time for there to be a blood flow response. Right? Some area of the brain has been particularly active, therefore it's hungry, it needs more blood. There's a hemodynamic response that delivers oxygen and glucose into that region of the brain, and that takes seconds to happen. And so the interaction between fields of neurons we would expect to take place somewhere in between the tens of milliseconds and the seconds range, but actually, sometimes we can see interactions between fields of neurons taking place virtually instantaneously. So there's something that has to be explained there. Also, if we're really thinking seriously about the brain, we need to take into account other aspects of the brain's evolution. Okay? On an evolutionary time scale, the brain develops basically from the inside out. So the limbic structures that we have inside of our brain is the most primitive. We share it with like sharks. And the neocortex, the thin layer of cortical tissue on the outside of our brain, is the most recent evolutionary development. And that we can trace and see differences in the behavior and properties of neurons as we look at the brain in that anatomical scale. Also, we can see neural growth taking place. Now, in the case of infants, we can see the brain growing and developing, and it comes by the curling of the neural tube, and this allows certain types of interconnections to take place, which are special to the way that the brain works and the way the brain develops. We also know that neurogenesis is something that never stops. So, unlike what you've always been told, it's not true. We do continue to make new neurons. It's just that we're not very good at putting them to work anymore once we get them. So we've got scales of dynamic organization in the brain. We have neuron-to-neuron communication. In this schematic, we're looking at this is a drawing that outlines the pyramidal neuron, which has long connections, right, reaching up through the cortex. And it's interacting with stellate nuclei, which actually don't have these long reaches. They interact locally with the pyramidal neurons. And it's the interaction between these two. This is an excitatory neuron. This is an inhibitory neuron. And the interaction between the two gives rise, under certain conditions, to what we call gamma oscillations in the brain. And we think that this is basically the driving force of the oscillations that we detect in the brain. The little engine is right here. When we look at the mesoscopic domain, this area where I said we sometimes see instantaneous phase locking, what we're looking at really is columns of neurons, right, which give rise to electromagnetic fields, which propagate across the surface of the brain and through the thin tissue, and we can see its properties distributed in terms of sort of phase maps. And then at the global level, we've got these long connections which take information and move it around the brain. And this is what we're looking at when we're doing MRI scanning. And when we're taking steady-state electroencephalograms by measuring it off the surface of the skull, we're also looking at the global behavior of some neurons. Okay. So one of the interesting things about the brain is its connectivity. So in this picture of a neuron, we've got orange dots. This is a staining procedure where the orange dots actually show points where other neurons have connected to this one. Right? So you can see that the connectivity across the surface of the neuron is pretty high. And in fact, we figure that the connectivity in the brain is something called it's a small world connectivity, and it is there's about 10 to the 10th neurons, but there's many, many, many more than that connections. They're organized into columns, and these are the kinds of things that we're interested in when we're looking at these waveforms that we're going to measure. So what's the basic nature of neural computation? Well, it's time-dependent, it's parallel, and it's dynamic. So basically, all of those neurons are doing this. They have some kind of what's called an action potential. Information comes in and stimulates the neuron until it reaches some firing threshold. It then discharges, and it comes back to its resting state. And when it does that, the signal is propagated out to the other neurons that it's connected to, and they then may be triggered to fire, and they will connect to others. So this is just spreading phenomena, but there's time lag, and there's time lag, and there's competition for the resources. The neurons themselves actually need food to do that, and they compete with each other in order to succeed. So it's not just simply a linear burst. It's actually a highly structured one. Okay, this we've seen before. All right. So we've looked at neurons. Let's now jump to the higher scale. We can look at the blood flow and metabolic activity studies by using something called magnetic resonance interferometry. MRI uses radio frequency signals produced by displaced radio waves in the magnetic field. So you'd have a gargantuan donut, which is an electromagnet. It's superconducting, so that means it's bathed in liquid helium, and you stick somebody's head in it, and when they're in this very high magnetic field, all of the energy patterns of the water molecules in their brain all line up. Then you turn the magnetic field off and they relax, and you can actually... That's interesting. Oh, there we are. We're back. And you can actually then do a bunch of very fancy math and reconstruct soft tissue patterns. So it has certain advantages. There's no X-rays or radioactive material used, which actually sets it apart from many of the brain imaging methods that had been developed prior. It gives us a detailed view of soft tissue, which is really important. Right? It's safe, it's painless, it's non-invasive, and you don't have to do any kind of fancy special preparation. Its disadvantages are it's expensive, so I just bought one. It cost me £1.5 million. It can't be used with patients with metallic devices in them, which actually excludes a lot of people. So if you've had a cochlear implant, you can't go in a brain scan. Right? It can't be used with uncooperative patients because you have to lie still. And that's another issue that comes up. So people often say, well, why aren't you doing, for instance, work on Parkinson's patients? Well, only if you can get them to not shake when they're in the scanner. Why aren't you doing research on, attention deficit disorder children? Well, only if you can get them to lie still in the scanner, right? So there's a problem with sort of just a general methodological problem that has to do with movement artifacts. It's not very good if you've got claustrophobia. It's a little narrow tube, and if you're not claustrophobic when you go in, you're likely to be claustrophobic when you come out. And furthermore, it has really poor temporal resolution. Okay, so although it gives us beautiful pictures of the soft tissue, it can only do it in fairly slow, sluggardly time scales, which means that we've got limited to this type of questions that we can ask. But we do get beautiful pictures. So here's some pictures that we've generated recently from our scanner. This is actually different areas in the brain that are engaged specifically when they're doing numerical manipulation as opposed to linguistic manipulation. So we get a nice view of different regions which are contrastively engaged if you control your tasks in a nice way. You can slice and dice the brain any way you want it with this methodology, so you can look carefully into different regions and target areas of the brain that are of interest. Another thing that you can do with this is something called diffusion tensor imaging. This is a way that allows us to track connectivity in the brain. So if you're trying to understand what particular areas of the brain are connected to others, if you've done a study like this and you think, okay, this region of the brain here is of particular interest, and I'd like to know what parts of the brain this area, when it's active, is communicating with, then you do this type of imaging. And it allows you to track the connectivity patterns between a particular region, which was called the seed, and other areas of the brain. So this allows us to actually find connectivity patterns. It doesn't give us causality, so we can't say that this root causes that one to work, but it does tell us that certain regions are preferentially talking to each other, or at least preferentially connected to each other in a physical sense, right? So these are patterns that are actual physical connections. You're tracking the communication between water molecules moving along axonal tracks. Another thing we can do in this device is something called NMR spectroscopy. So we can ask questions about whether or not certain chemicals, protein complexes, are present in different concentrations in particular regions of the brain. So here we've picked out this particular chunk of the brain, and this is a spectroscopic assay. So it allows you to look, for instance, at the effectiveness of drug therapies. Is the drug getting to the area that we want it to get to? But it also can help us understand, for instance, if we're particularly engaged in a topic, or we're particularly bored by somebody who's going on and on and on, are serotonin levels in specific regions of the brain being elevated. So to bring this back now to language, in a series of studies, we looked at something called displacement in language. So in lots of languages, when you form a question, you do it by essentially pronouncing a question word which is understood to be connected to some place in the sentence that comes up much later. So in English, we say, who did Mary kiss? So that word who is not really interpreted until we get down to the verb kiss. So this is often described in terms of movement, but basically it's a delay of information. Here we can see again this standard pattern. All right, so what we're looking at is specifically the regions of the brain which are activated when we make these types of links, where the question word has to hang on until a constituent analysis has been completed such that we get an interpretation of it. And we see that we get activation of Wernicke's area, and we get activation in Broca's area. So just as the old picture suggested, Broca's area and Wernicke's area are engaged when we have this type of dependency going on in language production and language understanding. But languages don't only do that kind of stuff. So this study was done in Danish, and in Danish we get to do something called negative scrambling. What happened to my, there we go, there's negative scrambling. So in this case, under certain conditions in Danish, a negative particle which ordinarily is interpreted inside of the verb phrase, is actually pronounced outside of the verb phrase. So it's another kind of displacement, but it's not the kind of displacement that we saw before, where you've got a question word and you're waiting for its interpretation. Here what we've got is a negative particle, and its influence on the interpretation of the verb phrase is delayed. And this thing calls up activation in a different part of the brain. So remember before we saw activation in Broca's area and activation in Wernicke's area, and now we're seeing activation in the anterior temporal horn. This is an area that's been identified by lots of other researchers as well as being engaged when we do certain types of language tasks. So it's not a huge surprise that it's there, but what's interesting is that in the same language, we've got two different kinds of dependencies, okay, question word dependency, neg movement dependency, and in the same individual, they actually bring up quite distinct cortical activations. So we can look, using this technology, we can look at the brain and ask questions about, are there really distinct brain responses that correspond to distinct linguistic observations? So we believe that question constructions are different from negation constructions. We've got good reason to think that. There's a long history of linguistic analysis which supports it, and now we've got evidence in some Danish brain that suggests that, in fact, it's true. At least there's a physiological reality that corresponds to the linguistic generalization. Okay, so what we're going to do now is move back to this intermediate level of investigation. So we look now at, we've seen what we can do with the MR scanner. It looks at the global properties of the brain and the kind of question that we can ask. Is there a difference between, say, WH constructions and negation, and are there brain areas that correspond to that distinction? And now we want to take a look at this mesoscopic region, so the neuronal assemblies. And there's a famous guy in the field, a fellow named Walter Freeman, who's done a lot of the seminal work on understanding the mesoscopic processes, and this quote from him says, even casual inspection reveals continuous widespread oscillations. Basically what Walter Freeman is saying is he doesn't believe that it's a good idea to look at particular regions in the brain, because when we look at the brain actually doing brain stuff, what the brain does is oscillate. So let's pay some attention to the oscillations. So that's what this picture is about. Here we have the inhibitory neurons and the excitatory neurons all getting together and having a party and giving rise to this type of wave oscillating behavior that we can measure and see on the surface of the cortex and throughout the brain. I keep saying surface of the cortex, but it's not only there. And the way that we look at that is with a technology that's actually old and cheap. So it's called electroencephalography or evoked response potentials. And it kind of fell out of favor because it was old and cheap, right? Much cooler to do research that costs a fortune than to do research that doesn't cost a fortune. But it turns out that this technology can do some pretty amazing things if we pay very close attention to what the brain is actually doing. So I'll tell you a little bit about that. Evoked response potentials are a change in electrophysiological behavior in the brain as it responds to a particular stimulus, okay? And it contrasts with something you might be more familiar with, which is called the electroencephalogram. The electroencephalogram is what if you've been suffering from migraine headaches and you have to go into the headache clinic and they put things on, they leave you there for a while, they're collecting your electroencephalogram. ERP measurements, the evoked response measurements, are time-locked to the onset of a particular stimuli. And the measured contrasting components in the brain are labeled with respect to the voltage and polarity, whether positive going or negative going, with respect to after the stimulus has happened. So there's things called the N100, which is a negative going deflection 100 milliseconds after a stimulus is presented, and there's a P300, which is a positive going waveform 300 milliseconds after the stimulus was presented. You all just had a P300. So the P300 is elicited when someone does something surprising, like smack the table. It's a surprise response. The N100 is actually a detector of any type of stimulation. The P300 is something that the brain does when something unexpected happens. The information that we can extract when we look at these things includes latency. So when do these components happen? The early components, those are turned up between 0 and 100 milliseconds, are known to be associated with physiological responses to stimuli. They're robust, and what that means essentially is it's very hard to move them around or change them. You can't alter people's responses by giving them extra information. These early components are just there. Later on, right, as you move from 200 milliseconds downstream in time, different cognitive variables can influence the type of component you can see. So this surprise response that you just had is a response that can be modulated if I just keep doing it, right? You don't show the surprise response when it stops being a surprise. And if I tell you I'm about to bang my hand on the table, you'll give a weaker P300 than if I do it without giving you any warning. The time resolution of these things is actually dependent upon the equipment. So this conventional time resolution was 250 hertz. That means that we could sample the brain every 4 milliseconds. Nowadays, we sample the brain every 1 millisecond. There's been a change in technology. The higher sampling rate just basically generates bigger data files. And it used to be expensive to have big data files, and now it's not so expensive to have big data files, so we can sample at higher rates. It's not really a good thing. Sometimes you just got useless oversampling. And what we're measuring is the synchronized activity of assemblies of about 10,000 pyramidal neurons. And we know that because we can do some physics that will tell us that in order to be able to detect activity from outside the skull on the surface of the skin, there has to be at least 10,000 neurons acting in concert to generate something that we can actually detect. So the EEG, 10,000 neurons, and its voltage range is about 1 to 150 microvolts. So that's the thing that we're trying to detect. For the evoked response potentials, like P300, same amount of neurons, but the voltage range is a tenth of that, so 0.1 to 10 microvolts. So there's an interesting technical challenge, which is how we're going to identify these things. The way we do it is we put electrodes on the surface of the scalp, from 2 to 256 electrodes, and we then measure voltage generated over time at these different spots. Nobody does 256 electrode recordings because it takes too long to put them on, but it is a very macho thing to do. So it's sort of the testosterone aspect of the research is reflected in the number of electrodes that are used. Anything above 128 channels is kind of impossible to interpret because you get leakage between the electrodes themselves. You get volume conduction. Right? But it's often very cool to say that you did this 256 electrode recording. And the general setup is this. You've got some victim who has electrodes glued on their scalp. The information goes to a digital amplifier. The stimulus is presented to the subject, and we chop up the output of the amplifier into chunks that correspond to the auditory stimulus, so we know when the stimulus has been presented, and then we wait some time, we grab that. Next stimulus is presented, we wait some time and grab that. And we glob it all together and average it and do some filtering, and we get these components. Here's the math. I won't go through it. The paradigm that I want to show you is some more linguistics, but we'll do it fairly quickly. It's not too nasty. It's called, there's things that all languages have, and they're called polarity constructions. Okay, so we have things like negative polarity item is a word in English like ever. The research I'm going to tell you about was actually done in German, so that real stimuli are here. German jemals is like English ever. So we can say things like no man is ever happy. Okay, it's an all right thing to say. You can't say a man is ever happy. So this word ever needs to be associated somehow with negation in order to have an interpretation. And we have things that are similar that go the other way. In German, the word is durchaus, and the closest comparable word in English is certainly, so no man was certainly happy here is a strange thing to say, as opposed to a man was certainly happy here, which is an okay thing to say. So we've got a word that requires association with negation, and a word that requires association with something positive. Okay. These are interesting guys because they're not predicted by their grammatical context. So no man is ever happy is okay. No man is happy is also okay. The existence of negation doesn't require the existence of the polarity item. And also, the category itself doesn't tell you that it's going to be a polarity item. So no man is casually happy is the same part of speech as ever, but it's not a polarity item. Just to make it more interesting, it's not enough that there's something like negation present. It has to be in the right place. So it actually has to be structurally in the right place as far as in the sentence for it to work. So no man who has a beard is ever happy is fine. But a man who has no beard is ever happy is not good, even though there's no in the sentence. Right? So here's no here, there's ever, and this is okay. Here's no here, and there's ever, and this sentence is no good. And the reason is that this negation here is actually stuck inside of a relative clause and is inaccessible as far as this polarity item is concerned. The positive polarity items and the negative polarity items present the same type of processing challenge. We don't know they're coming until we get to them. And once we get to them, we have to recognize that they're a polarity item and then make some kind of analysis to determine how we're to understand the sentence. So we don't expect there to be any real difference between the two. When we want to put the ERP approach to work then, we've got some tools that we can work with. And these are these components that we can see in the brain. And with respect to language processing, there are several components that we know or can be reliably elicited. And we're going to look at two of them here. One is called the N400. It's a negative going deviation, peaking typically around 400 milliseconds. And it is a measure of semantic integration. And let's look at an example of it. So the N400 is sensitive semantic anomaly or plausibility. If you've done psychology, it is a close task identifier. Okay? Here's my sentence. The knight in shining armor drew his sword. Okay? I've done this all over the world. It doesn't matter what your language background is. Everybody says sword. People who only speak Tamil say sword. Okay, so knight in shining armor drew his sword, but you could also say the knight in shining armor drew his blade, right? Blade and sword are pretty good. And in English, you could say the knight in shining armor drew his pay. He must have got paid. Okay. But what happens when we do these recordings is we discover that this N400 component, right, turns up to have its duration and amplitude reflecting the goodness of fit of these words. So here we're plotting time, and here's the onset of the word, either sword or blade or pay, and we count 1, 2, 3, 4, and there's a little bump. Negative is plotted up in this by convention here. So here's this little bump, and this is the N400. You always get one. It shows you that you're integrating this word sword into the sentence. And this red line shows the cost of integrating blade, which is bigger. And here's the green line, which is the cost of integrating pay. So the N400 is a nice marker that shows us the effort that's being used in order to integrate these things. We have another component called the P600, which is a positivity, obviously, peaking around 600 milliseconds. And it reflects the cost of syntactic reanalysis and repair. So in sentences like, while Mary dressed, the baby played on the floor, elicits a P600. While Mary dressed, the baby, right? Here the baby is either the object of dress or the subject of this clause that's coming up. And people tend to interpret it as the object and then discover that they're wrong. In German, you can do it in a much more simpler way. Welke Frau is ambiguous between nominative and accusative case. The verb agrees in number with the subject. Okay, so here's Welke Frau as the subject of the sentence. And we know in all languages that we've looked at, people assume that the initial noun phrase that they encounter is the subject of a sentence. So the interpretive strategy is which Welke Frau is the subject here, and then they get the word za, okay? And in that case, it's singular, and so is Welke Frau, so everything's fine, okay? However, if they get the word sein, Welke Frau sahen die Männer, which is okay, another okay sentence of German, this is plural, so this can't be the subject. This is the subject, so you have to change your mind, and that elicits this positivity. Okay? So this is the P600 component. And so we have a prediction. Failure to license a polarity item should elicit both an N400 and a P600. And these are our sentences. A man is ever happy, which is bad, and polarity items are sensitive to their meaning, environment, and therefore we expect an N400. It's going to be very difficult to interpret this element in this environment. There's no negation, so we expect the N400. And also, a man who has no beard is ever happy is bad. Polarity items are sensitive to the hierarchical constituency, This is a negation is there, but it's in the wrong place as far as the structure of the sentence is concerned, and therefore we would expect a P600. So we did it, and I want to remind you that a licensor has to be present and a licensor has to be accessible, so there's both a semantic or a pragmatic condition as well as a structural condition. And also, I want to point out that from the grammatical point of view, and also from most conventional sentence processing theories, the relative clause here plays no role in deciding whether or not this sentence is good or bad. A man was ever happy is bad, and it doesn't matter whether there's negation or not in the relative clause, it's still bad. And a man was certainly happy is good, and it doesn't matter if there's negation or not in the relative clause, it's still good. So the stimuli were these types of sentences, and these are central electrode recordings. And what we see here is that the blue tracing is a conventional no man who had a beard was ever happy. So negation in the right place, no other negation, and there's the negative polarity item. A man who had a beard is ever happy, no negation at all, and it's bad, and that's this red tracing. And when we have negation in the wrong place, we still get this N400 component, right? So here's an N400 component, and this is a P600 component. So we do get an N400 and a P600, but we actually see an effect of having negation in the wrong place, unlike what the theory said. In this case, this is the positive polarity item, and here's the good positive polarity item, the blue tracing. The bad positive polarity item where we have negation, and that's the red tracing. And again, we get our N400, and we get a P600. And then the instance where we have negation in the relative clause, which is still a good sentence, gives us no modulation in the N400. So here we see it's an N400 that's modulated. Here we don't. Okay? And looks like we get some modulation in the P600. So the results support the prediction that there should be both an N4 and a P6, but contradict the theory which suggests that there shouldn't be anything there. Okay, so here we've seen some evidence, a way that you put these ERPs to work. And I see that I'm running out of time, so I'm going to snap up a little bit. I'm going to poke this analysis a little because I think it's important. So what we've just seen, this N400, P600, and the P300 demonstration that we did earlier, are all components that are identified by looking at electrocortical activity in the brain and averaging it. Okay? Now, an averaging technique is a linear technique. The brain doesn't actually behave in a linear way, so there may be something wrong with it. And in fact, there's a bunch of things wrong with it. So we know that for the averaging technique to be true, certain assumptions are being made. One is that the electroencephalogram is an ERP, the evoked component we're looking for, embedded in noise, that the signal is invariant across trials as well as across subjects, that the noise is stationary and ergodic, basically means that no matter how you look at the noise, it looks the same. And the ERP and the noise are uncorrelated, and the noise across trials are uncorrelated. These are all not true. So we know for a fact that these are completely wrong statements, and in fact, everybody knows they're completely wrong statements. It's just that the conventional practice is to average. Okay? Now, what this means is not that these ERP results are unreliable. It means the converse. The fact that we get reliable observations out of these brain recordings using this method at all is amazing. So these are hugely conservative estimates of coherent activity in the brain. So they're not unreliable. They just vastly underrepresent the kind of information that's available using this technique. What should I do? Shall I finish this off? Okay. So bear with me. So what we're going to do now is have a quick look at a nonlinear approach to analyzing the same data. yada, yada, and here we go. So there's some other ways of doing it. Symbolic encoding methods allow us to take a look at the data in a way that's different from the averaging approach. And we can also have a quick look at synchronization. We're going to look at phase synchronization results. So if we try and take a look at we assume that this is the actual analog output of the ERP recording, so some sort of general squiggly line that represents changes in voltage over time. What we're going to try to do is change this information, which is continuous information that links voltage values with points in time, to some symbolic sequence of zeros and ones. And there's several ways of doing it. One is a baseline method, another is a uniform encoding method, and I'm going to tell you about the half-wave encoding method because that's the most powerful one that we developed some time ago. The way that you do the half-wave encoding is you take the analog output, okay? You choose a window of time. So remember, these are all observations of a voltage value at some point in time. So you decide on some window of time. So let's say we're sampling every millisecond. We'll take a window of time that's 10 milliseconds long. Inside, we start looking inside of a 10 millisecond window, and we ask the question, is there a negative going or positive going deflection of the waveform in that window? Okay. You do some smoothing algorithms and stuff, but basically that's it. You choose your time window, and you decide, does the waveform go up or does the waveform go down? And if it goes up, you give it a 1, you call it 1, and if it goes down, you call it a 0. And once you do that, you can start computing something called a cylinder entropy, which is basically you take that information, you pile it all up together. So for every trial, you look at how often you've got a 1 and how often you've got a 0, and you construct a cylinder which corresponds to a point in time. And on that, you can compute a value called entropy, or a version of Shannon entropy. Entropy is interesting because it tells you about the coherence of the signal. Is the signal doing more or less the same thing, or is it actually being driven apart? If we've got low entropy, then there's more ones than zeros, or more zeros than ones. So the signal is mostly doing the same thing. Maximum entropy is when there's as many ones as there are zeros. So it's a 50-50 distribution. Okay, so here's a plot of exactly the same data, only so this is the negative polarity construction. Here's the half-wave encoded version. And what we can see is some interesting differences. All right, so the plot is different. This is voltage, negative up, positive down, and this is bit sample. Time is the same. This is time this way, this is time. So one of the things that's interesting is that in the case of the negative polarity construction, we have this modulation of the N400 when there's negation in the relative clause or negation not in the relative clause. And what this says is that the brain response happened at the same time if the sentence was bad because there was no negation in it at all, and also if the sentence was bad because negation was in the wrong place. And that's a surprising result. You would expect there to be differences in time. If we look at the half-wave encodings, the entropy measures tell us that there's a decrease in entropy in the case where there was no negation in the string at all. It happens earlier than in the case where there is negation in the string, but it's in the wrong place. That makes a lot more sense. And the reason that we have that, actually, is because when you're looking at time and averaging, you're also averaging over time. So we kind of smear the possibility of getting these distinctions when we do an averaging technique. With the half-wave encoding, you actually hold time quite rigid. You decide how big your temporal window is, and it fixes how well you can discriminate. The most important thing, though, is this big blue drop. So what this tells us is that when we get to the word ever in a sentence that's well-formed, the brain response is to become highly coherent for a particular period of time and to stay that way. And that seems to be a reflex of successfully solving the problem of interpreting this string. The same thing is true in the positive polarity construction. Great big dip that corresponds to successfully interpreting the sentence. That's a surprising result, because if you, or not surprising, a very desirable result. And the reason is that if you think about the history of trying to understand the relationship between the brain and language, it comes from always looking at something's wrong and trying to work backwards from something that's wrong to say, well, if this is what's happened if someone's been shot in the brain, then, in this particular spot, then it must be that that spot had something to do with the fact that he can no longer dance the samba, right? And when we get to normal language processing, we take sentences and create things like the horse raced past the barn fell, or these crazy German sentences, negation in the wrong place, and you ask what people do when they're confronted with some sentence they're unlikely to ever see. Here we're seeing, but that's the only way you can trick the brain into giving you a response that you can measure. But here we're seeing that by changing the analytic approach, right, we actually start seeing systematic responses that are about the successful solving of a cognitive task, in this case, the successful parse of the string. So for both negative polarity items and positive polarity items, we get this sustained drop in entropy. We see this successful integration of information, and the nested effect turns out to be temporally distinct. Okay, so there's one last point to follow up, and then we're done. So when we take a look at negative polarity constructions of the type that we've looked at, they have something called a scalar meaning. So no teenage boy will ever come near my daughter means it's highly, highly unlikely that that's going to happen, okay? So ever tells us that that number of instances of that event is at the negative end of the scale, and if I try and push that likelihood away from the negative end of the scale, it becomes a bizarre or unacceptable sentence. So I can't say, no teenage boy will ever come near my daughter, but not often. Bizarre thing to say, okay? But the positive polarity items that we looked at don't do that. So a teenage boy has certainly come near my daughter, but not often, is all right. So there's a difference between the general properties of meaning that are encoded in these structures. And we can look at that further by taking advantage of these time windows, okay? So remember we said you take this, for the half-wave encoding, you choose time, a time window when you look whether or not things are going up or down in that particular time window. For the negative polarity items, we did a whole bunch of time windows, so here's just representative ones. If you change the time windows around, you discover that at 640 milliseconds, a window that corresponds to about 0.78 hertz in terms of frequency in the brain, you get the biggest deflection, right? So the biggest entropy drop, the strongest marker that the signals become coherent when the sentence is well formed. If you look earlier, if you look at smaller windows or bigger windows, this entropy drop is less. So there's something that, there seems to be a particular region, frequency region, which is associated with that interpretation. If you do the same thing for the positive polarity items, it turns out that it doesn't matter what time window you look at, you never get a difference in the size of the entropy drop. So the entropy drop associated with licensing positive polarity items is not sensitive to the frequency bands, but the entropy drop associated with licensing negative polarity items, these ones, okay, remember we can only talk about the examples that we've used, are very sensitive to the frequency bands. And once you know what the frequency bands are, you can actually look at the global behavior in the brain. So this is instantaneous phase synchronization, this is time, okay, this is frequency, and the color coding says there's more phase synchronization if it's red and negative phase anti-synchronization if it's blue. This is the negative polarity item, and we find in the low frequency bands very high phase synchronization. And for the positive polarity items, we don't find anything like that. It looks like we might get anti-synchronization in some spots, but it's patchy. So the application of these nonlinear methods allow us to do some tricks. It's possible to see cortical reflex of successful cognitive processing, which is a good thing. It's possible to discriminate the influence of linguistic content, maybe, from processing demands by carefully investigating the temporal dynamics in the time series. And it looks like timing counts and relative timing counts and time domains count, which is not a surprise if what we're looking at is oscillations as opposed to points in the brain. So the anatomical studies that we've talked about tell us about what physical areas of the brain are active and are differentially involved in language processes and other cognitive processes as well, these metabolic measures of activity. The temporal studies, we can use, say, the classical application of the ERP methodology tells us about general time course and the interdependence of language processing events in time and in the brain. And the dynamical system analysis tells us something about the properties of those systems that can live on that anatomy and generate that time series. So we've got us trying to sort of close the circle by presenting a way in which we can make use of anatomical information, temporal information, and dynamic systems analysis, and take advantage of our special knowledge from linguistics, which lets us look at funny sentences like polarity constructions, and learn something new about the relationship between language and the brain. And that's it. Thanks very much.
Thanks very much, Doug, for a very interesting talk. I guess one advantage of hearing an interdisciplinary talk is that everyone can get a message from it no matter what the background is. So this is rewarding. The bad thing for you is that you have to give double background when you're presenting and therefore spend time. So it's really great to hear that. So I was wondering if there are any questions from the audience for the talk. Jonathan.
Two very quick questions. One, I apologize in advance. I'm sure you've been asked this by every time you address linguists, but I can't resist. Have you compared the WH movement effects with languages that don't have WH movement? In other words, languages that use in situ?
Right. No.
Okay, that would be interesting to see.
It would be very interesting. We're hoping to do that. The problem is it tends to be, actually, so I do have projects going with colleagues in India that are starting to look at that now. The problem is basically technological. It tends to be the case that the languages that have in situ WH constructions don't have ERP or brain centers. Now, Japan is a different place. So there is research in Japanese.
Yeah.
And that suggests that you still get Broca's area activation. I mean, there's this article by Josef Brzezinski that tries to pull all of these observations together, but he's arguing that Broca's area is syntax, and I don't believe that at all. But
okay, because I was trying to see if you could actually tease out that it is not the processing of a normal WH question, however, whatever the syntax is, whether it's specifically reflecting the movement part as opposed to just the WH part.
Well, you'll know it's hard to make the case because even in the WH in situ languages, there's this assertion that there's still potentially covert movement going on.
Yeah.
So it's the problem of the linguistic.
And the second question was just one for comprehension. When you were giving your examples while the mother dressed the baby,
right.
Were those written stimuli or spoken?
Both.
Both?
Yeah.
Because what I was trying to understand is it would appear that the spoken should work quite a bit differently since you will have audible distinctions between, that the misanalysis would be at least less likely in the spoken than in the written.
It's the results. What Jonathan's saying is that there's a difference between reading and speaking, and that when we speak, we use our intonations to convey the meaning we intend. So the expectation would be that when we confront sentences that are ambiguous, like, well, the mother watched the baby played in the crib, where the baby could be either an object or a subject, we wouldn't have that trouble because when we hear it spoken, there'll be intonational information which will help us. It turns out that the answer, that the reality is more confusing than you'd expect. So the good news from, I'll put a particular perspective on Jonathan. The good news from Jonathan's point of view is that you can make people reanalyze normal sentences by giving them screwy intonation. Okay? So it's clear that prosody, intonation patterns, actually do push you to do things that you wouldn't ordinarily do. The other side of the coin is surprising in that the intonation patterns in ambiguous strings like this one don't perfectly disambiguate them. So it's a lot like the lexical ambiguity story. It looks like we still recognize that there's an ambiguity there, even though there's intonational information that tells us otherwise.
I actually had a question that sort of bears.
Yeah, but you can't. There's a proper question from that.
Oh, okay, sorry. Didn't hear it. I apologize. Yeah, go ahead.
Thank you. I was wondering whether your nonlinear method could be applied to second-order potentials like mismatch potentials, like different waves, and whether it could be useful to do that, in your opinion. Thank you.
So, yes, there's something called the mismatch negativity, which is a brain response, which is very interesting. It happens very quickly. I have an N100. Oh, so 100 milliseconds after we hear a stimulus, there's this negative going peak. Okay. If we hear a series of similar stimuli, so say a series of tones, and then a deviation from it, we get that N100 actually changes its shape. It shows us a larger, it gets slightly higher and slightly longer in its duration. And this is called the mismatch negativity. It tells us that we detected a deviation from something we're taking as a standard against something that's coming in that's not matching to that template. The reason that the mismatch negativity is really interesting is that it can be used to probe all kinds of categorical perception. So, for instance, you hear me speaking English, and you hear me saying the letter B, okay? Now, I say B in a very particular way. I grew up in Vancouver in the Pacific Northwest. Then I went and lived on the East Coast of America, and I've been all over the place. And the way I pronounce B's is probably unique to me due to these strange influences. Many of you aren't native speakers of English, so your notion of what an English B is likely to sound like is going to be particular to you and your own language experience. And yet, when you're presented with a series of tokens of B's, which range all over the place, you all say, it's a B. You don't say it's something else. The mismatch negativity can be used to find out exactly when something like this stops sounding like a B to you. And it turns out to be a very interesting question, because this is exactly what infants have to learn when they're acquiring the phonology of their language, of their mother tongue. We all come into the world able to learn any language. We come into the world able to make any type of discrimination. But in the languages that we come to speak, there's going to be lots of distinctions which aren't attested. Okay? At some point, we come to be able to recognize the proper categorization for our own language, and we behave in a particular way, such that we will recognize the candidates for B, if we're English speakers, whether they're pre-aspirated or post-aspirated, because it doesn't make any difference in English whether you have pre or post-aspiration. But if we were speaking Khmer or something, it would actually be two different consonants in the language. So we can use this mismatch negativity to try and pull this apart. Now the problem is, mismatch negativity is extremely small, right? And it's a delicate thing to look for. So that was a very long response to say, yes, using alternative data analysis methods do help us sharpen up the mismatch negativity, but it kind of pulls it apart. So when you move away from the conventional analysis, you always have the problem of saying, okay, do I care that the results I get using a different toolkit, which make different assumptions, deviate from the conventional wisdom? All right? And that's sort of the sociology of the field, and you have big fights with people about whether you're showing anything useful.
I understand it's possible to measure a sentence we are talking. Is possible to measure the sentence we are thinking?
Good question. Very good question. Very, very, very good question.
Identify, huh?
Okay. Not yet. Not yet. But there is an interesting take on that. So this N400 component, which I showed you, which is the knight in shining armor drew his pay. Right? You get them for all kinds of silly constructions, like I had a drink of rocks. Okay? That gives you an N400. However, if I first of all tell you a long story about I've just discovered this wonderful new Slovenian drink and it's called rocks, right? Isn't that funny? And then I say I just had a drink of rocks. No N400, because I've set it up such that the label rocks is going to apply to something you can drink. People who have suffered brain damage and have something called locked-in syndrome, and also people who are comatose, can show an N400 response. And this is used in some of the protocols that determine whether or not you're going to turn off life support systems in some cases. So you can if you are unable to respond, your brain can still show whether or not you're processing the language, because it can give an N400 response when you hear, or you're presented with an expression which is deviant, like I had a drink of rocks, or I buttered my canoe, things like that. So we can't see what sentence they're thinking, but we can make use of these phenomena to look into the activities of brains in people who otherwise can't communicate very well.
Hello. I'm not into linguistics, but I really liked your lecture. And I have this question about the graphs you showed, negative and positive polarity items.
Do you want to put them up?
No, it's okay. I just want to know, you said that you averaged some values. So was the experiment done on one person, listening different or speaking different statements? Mm-hmm. Or it is done on different people and then you averaged the results to get this graph?
Good. Very good question. The conventional methodology for ERP paradigms is group studies. So what you'll do is you prepare a whole bunch of sentences like a man is ever happy, right? So a man is ever climbing Mount Everest, right? All these things that are bad, and a whole bunch of ones that are good. So when I say a whole bunch, it's around 40 examples of the bad ones and 40 examples of the good ones. And then you test around 20 people on these things. The reason is that what you're trying to do, one of the things you're trying to do with the voltage averaging method is improve the signal to noise ratio so that you can find this very weak signal embedded in all of this other activity that's going on in the brain. And so you get people to do the same decision or hear the same token many times, and you do that to many people, and then you do the averaging. Yeah, you do some other things too, but yeah.
Are the sentences that they hear taped, or there's always a person saying it?
They're taped, right? So
okay,
so
it's the same.
So you either do it visually. If it's visually presented, then it will come in phrases. So it would be, a man is ever happy, presented word by word on the screen, or else on a tape that will be presented, and they're interspersed randomly, so that you don't get a whole bunch of the same type of sentence in a row. And there's filler sentences, so you don't only hear polarity constructions, you also hear other types of sentences.
Can I ask another? You showed that some results of the study that was done in English, the study in German, and the study in Dutch language.
Danish.
The third was?
Danish.
Danish, okay. So I happen to know something about these languages, and I know that the grammar has a different structure in each of them. So are there any correlations between the studies that are done on different languages, on native speakers, or it's like a bunch of things that you find can be correlated only for one language?
Another excellent question. Mostly we find that languages behave the same. There are things that are language particular, though, and they sort of have to do with sort of systemic properties of the languages themselves. So, I'm trying to think of an example. So all of the languages that create questions the way that we do in English, the Germanic languages do, show these types of things that I showed you. So, and we would expect it to be true of any language that creates questions the way that Germanic languages do, which is lots. The way that relative clauses themselves are processed, though, can be different from language to language. And there you get sort of idiosyncratic processing profiles. So in German, even though the German grammar doesn't require case matching between the head of the relative clause and the relative pronoun, German speakers behave like it is a requirement. I apologize to the non-linguists in the audience. It won't mean much to you. But if German speakers behave like there's a requirement in their grammar that isn't really there, English speakers don't do that at all. And it seems to be because German has an overt case system. And since there is an overt case system, they actually have processing strategies that make reference to an overt case system. English speakers don't have an overt case system, and so this particular strategy doesn't arise.
And just, I would like to know, is there a difference if you do a study on a native speakers population, or you do some studies with people who are not native speakers? And is there a difference in these negative and positive polarity items in people who are polylinguistic, so who speak like 5, 6, 7, 8, 10 languages? Thank you, that's all.
All right, okay. Yes, there are differences. So monolinguals behave differently than bilinguals or multilinguals. People who are multilingual or bilingual who are tested in their non-dominant language look different than they do in their dominant language, and they also look different from monolinguals in that language. So if you're, we're actually doing research on this in Basque and Spanish. Okay. So everybody who speaks Basque speaks Spanish. There are no monolingual Basque speakers, despite what Eta might want to tell us. But there's plenty of monolingual Spanish speakers. So you can look at this distinction in between Basque and Spanish. And what you do see is that these ERP components differentiate between first language and second language. You can ask the same question about language development. When children are learning to speak, they don't show this P600 component. They also don't show something called early left anterior negativity. So they don't show the components which seem to have to do with restructuring constituents, but they do show this N400 component fairly early on. So we see that in children who are learning language, a strategy that looks similar to what we see in people who are learning a second language, and that is you rely on your guess about the interpretation of the second language. You show N400 components a lot, and it's only when you get to a certain level of competency that you start seeing this P600 component coming in. In the case of bilinguals, people like me, for instance, who learned their other languages after puberty, you never develop this early left anterior negativity. This is a very early component which shows that you're making a judgment, a guess about the constituent analysis of the string as you're hearing it. You're making a very rapid assessment of the structure of the sentences coming in. You do that in your mother tongue, and you never get it in your second language. People who are raised bilingual, so they're born into a mother speaks A and father speaks B, and they live in the community where A and B are both spoken, they can have ELADs in both. But that's, you know.
That was first, I think. Oh, right.
I have a general question. Does the capability of mastering many languages depend on any characteristic of brain?
I'm sorry, I didn't quite understand what you said.
Does the capability of mastering many languages depend upon any property of a brain?
Not that we've seen. Okay, so it does seem to be. It's hard to say. There hasn't been enough research done on real multilingual communities. So, being monolingual, right, is actually quite rare. It just happens that there's lots of rich monolinguals. But generally speaking, being monolingual isn't the norm. So we often see people who have 2 or possibly 3 languages, but there are also people who have like 6, 7, 8 languages. When you look at language processing in these individuals, it doesn't seem to make a hell of a lot of difference, right? Which is kind of a puzzle. So there seems to be a first language, and then there's other languages, and you can't seems that the evidence suggests that you can have many other languages, and not an awful lot discriminates between the brain responses to the other languages. There's mother tongue, and there's other stuff. That's probably, it could, so first of all, I don't know all that much about it, so I may be just completely lying to you. Second of all, there's just not enough data, really. There hasn't been enough invested in this task. And there's good reason. I mean, I've been trying to convince the British government to spend money on this because England has become quite a multicultural place, particularly in the Thames Valley where I live. But the speech and hearing services for people who suffer strokes is almost entirely for Anglophones, right? And for the people from the Indian subcontinent who live in the area, their rate of stroke is higher than the rate of stroke in the rest of the population. So there's a lot of people who are suffering brain damage who are not benefiting from language assessment and language therapy because in England we speak English. So I think that this is an area that really needs to be addressed, and the European community is going to start spending some effort dealing with this because it's not at all uncommon.
I think there was one question in the back here and one here, and then I think we'll have to stop and let that rest.
I'd just like to know what are the applications of such analysis in lie detection?
There's an awful, okay, this is a controversial issue. There's a lot of claims made about the ability to use these brain imaging technologies for lie detection. I think it's mostly unreliable rubbish. There's a thing called negative contingent variation, so there is a component in the brain which seems to be elicited when you are about to answer a question wrong. Okay, so if you have a task where you have to study, right, and then you're going to be questioned on it, you can see in the brain record a particular type of electrocortical activity which tends to co-vary with the likelihood of making an incorrect response. Okay? And that, on the basis of that, there's been a lot, I think, on the basis of that, there's been a lot of claims made. Now, most lie detection approaches don't simply rely on negative contingent variation. They also do electromyography, so skin conductance tests, heart rate, and all kinds of things. But I'm still skeptical about the reliability of it.
One here.
My question is, in a way, very simple, but I have been thinking about how to put it all this time. Basically, what my question is where do you see the bridge can be made between language and brain function? To put it in context, the linguistics know a lot about the organization of language, right? How sentences are made, what makes a good sentence, etc. From the viewpoint of semiotics, we can even tell about how meaning in a way is organized, etc. And we know it's a very complex system in itself. And for me, this can be a problem because we know so much about language in itself that we tend to get the idea that there is some kind of direct representation of that complexity in the brain. But that, in a way, that presupposition could be problematic. Because, on the other hand, cognitive science in general, let's say when you use imaging techniques or whatever, you enter with an idea and you say, okay, I think memory works like this, and then you look at the images and you see, well, maybe it's not true, maybe it works like something else, and you have room for improvisation. But here in the case of language, because it's so well defined inside linguistics, there is really not so much, if you see what I mean, room for improvisation, because if it's not that, then what is it? So my question is there a way to maybe step out of the understanding of language to find a new language? So simply, how to put that bridge if it turns out that it's a completely different grammar on the level of...
Okay. This is an important question. Okay, so I don't blame you for having a hard time articulating it. And it can be answered in a variety of different ways, and it sort of applies to a bunch of different issues. So there's a question about what constitutes evidence. So if you've got a theory of language that says, all nouns are quantifiers or something or other. Right? So you've got some claim that's a bit slightly controversial, but you've got an entire edifice built around this property. What kind of data would actually make you revise that particular position? I mention this because Bertrand Russell thought this, and he never changed his mind. So what would, and I'm very sure that Bertrand Russell would not have changed his mind if I showed him some pictures and some graphs. I said, look, here's nouns that are clearly not behaving like quantifiers, because, and then I'd have to give him this extraordinarily complicated story about brains and signal analysis and stuff, and he'd just tell me to go away. Which is essentially Chomsky's approach too, right? So I talked to Noam about this stuff, and he's excited and he's impressed and he likes the points of contact, but he's not very interested in the points of non-contact. So there's a question of what constitutes evidence, right? And also what constitutes evidence in the other direction, right? So if I assert that there's some region of the brain that's particularly supports some type of linguistic process, and then you come along and provide me with an example from Slovenian, which looks like it has to be that particular process, and in the brain it doesn't engage the area that I say, what am I going to do? Well, I'm going to tell you to go away and revise your analysis, right, about this particular linguistic analysis. So there is no easy way of putting these two things together when there's a general disagreement. However, there's a different way of approaching it. And the approach that I take is to ask the question, how much of our descriptions of language, for instance, are unnecessarily complicated? What guides me when we're doing our research about understanding how the brain works is trying to understand the physical principles which underlie the function of the brain. So what aspects of the physical property of the world that we live in determines the kinds of things that we see going on in the brain? So, for instance, the fact that we get this gamma burst because of this interaction between the excitatory and inhibitory neurons, which have different kind of scales, that is something that can be explained because the physics of the world is that way. We do not require there to be genetic encoding that makes that happen. We don't have to imbue people with any type of special properties to get this type of phenomena to come out. So one of the questions, one of the leading approaches is to try and understand those aspects of the physical world which determine the phenomena that we see in the brain, so that we can tell the difference between those things which we really require an explanation. That is, we have to rely on genetics or proteomics or metabolomics to explain why the phenomena we're looking at is such and so, as opposed to things that would happen anyway. And when it comes, and that's the same approach that I take to grammar. I think that we tend to complicate our descriptions of human language by imbuing them with all kinds of machinery that we think are linguistic machinery, but they don't have to be. They are properties of the way signals work, or they're properties of the way that things like non-complex systems behave, right? The fact that it's an information-based complex system doesn't mean it doesn't behave like a complex system. And there's properties that complex systems have, and we see them in language. And we also see them in the water going down the drain, right? We're missing an enormous opportunity, I think, by not taking advantage of the insights that we have from recent advances in mathematics and physics and engineering and cybernetics, which allow us to look at complex systems phenomena and bring us tools and means to investigate them, and not applying them to the complex phenomena that we're working on as cognitive scientists. And that's sort of the hidden agenda in my story.
Thank you. If you still have questions for Doug, I would encourage you to ask them during the reception period. I'm sure he'll be happy to continue the discussion, this very interesting discussion. So in the meantime, I'd like to thank you very much, Doug, for an extremely interesting talk and discussion. And once again, welcome to you to Slovenia.
Thank you very much.